hosting me. It's always great to be back in Kingston. As Jordan said, we both did our degrees at RMC. I didn't come to this side of Kingston very much, but it's like I said, it's always great to be here. So I'm going to be talking about things that I've been doing in my research for about the last 10 years. So in terms of my training, as Jordan mentioned, I'm an electrical engineer. I was trained in communication theory, so I did a lot of things in wireless. And about 10 years ago, I decided wireless is kind of, to be perfectly frank, I found it kind of boring. And I was looking for something a little more interesting to do. And I had this wild and crazy idea that maybe instead of communicating using electromagnetic radiation, what if we started communicating using chemistry? And so I thought this was the world's most original idea. It turned out that people had been looking at this for decades before I got on the scene, and I managed to plug into a pretty interesting and still growing and still developing area of research that touches on a lot of other areas such as chemistry, biology, physics, and so on. So I'm very happy to share that with you today. Just like to quickly acknowledge this is the work of many people, including students, collaborators from around the world. And I always want to acknowledge my funders, and more funding, please. So here's a bit about what we're going to be talking about. So the term molecular communication, in some sense, I guess, is self-explanatory, but it is a little bit different from what we normally think of when we think of communication. So I'll start by motivating that, talking a bit about what we mean by molecular communication, why it's important, and a quick idea of how we might communicate using molecules. I'll then go into a bit of a more theoretical way of thinking about molecular communication. So my background, as I mentioned, is in information theory. So we'll think about some information theoretic aspects of molecular communication, how it's modeled, and how it's conducted mathematically. Then we'll move into some case studies. So the first one has to do with some of my more recent research, which is considering how a study of molecular communication impacts on biological communication. So organisms tend to communicate using chemistry. That's what they have available to them. Not always, there are exceptions, but a lot of communication, particularly amongst microorganisms, is chemical in nature. And I propose that by studying molecular communication, we can actually understand a little bit about what organisms are doing when they communicate, and even make predictions about what they're going to do. The second case study is more on the engineering side. So I'll show you some things we built in the lab. So you might think, okay, molecular communication, this is chemistry. So maybe we need a big wet lab setup. No, in fact, I don't have a wet lab. Instead, we came up with this funny idea of, as mentioned, communicating using sprays of vodka. And it was a pretty fun project, and I'm looking forward to sharing it with you. I did write a book about this, so this is basically all of the things I'm about to talk to come from this book. On my lab webpage, there's a link there. I was invited a couple of years ago to do a short course. There's about 8 hours of lectures there, so it's just all available. If you're interested in learning more, please feel free. And there's my email if you want to reach out and ask more. I'm always happy to communicate with anybody about this. Okay, so getting into the meat of what we're talking about today, molecular communication. What is it? Why do we do it? And how do we do it? So what is molecular communication? Quite basically, it's communication using chemical properties. So if we think about how communication works that we're normally familiar with, I see many people with laptops, phones, and so on. You are communicating using electromagnetic radiation. Almost all of the engineered communication that we do is electromagnetic in nature. If you think of like an electric field or an electric potential down a wire, or electromagnetic waves or something like that, it's all electromagnetic. But in nature, as I mentioned, almost all of the communication that occurs, for instance, between cells in your body or between microorganisms is chemical in nature. So there's a huge amount of molecular communication happening in which we manipulate, in which the entities that are communicating are manipulating the chemical properties of a system in order to express a bit. Why would we do this? So there's really three reasons that have been sort of proposed in the literature over the last 10 years or so. Where this first came from was a solution to what's called the nano-networking problem. And this is like, there's like a trope in science fiction of like, in the future we'll have like nanorobots swimming in our body doing various things, such as targeted drug delivery at the site of a tumor, or microsurgery using these nanorobots that just go right to where they're needed and then flush themselves out. So that has been thought about. I mean, it's been conceptualized for a very long time in science fiction. It's been worked on in detail in labs for actually quite a long time. And I think the surprising thing might be that, I mean, this is obviously a cartoon, but devices of this size, they're old news. They've been designed in labs and used for very simple tasks for decades. There are at least 3 research groups around the world that build these devices, that build tiny little devices that are small enough to fit in the bloodstream. The problem is we can't get them to, like, one tiny little device can't really do very much, and we can't get them to cooperate with each other. An issue is that electromagnetic communication, for example, doesn't work well in the body. The body is salt water, and even if it was free space, the size of the antenna means the required size of the antenna means that you can't communicate efficiently. So some people looked to nature for inspiration for communication solutions to this problem. And it was thought that, well, if we could think about how how we can communicate using molecules, which is the solution that nature has found, perhaps we can come up with the solution to the nano-networking problem. So that's one motivation, is basically nanomedicine. As I've mentioned, another motivation is biological communication. So going the other way, rather than taking inspiration, why not take the analytical tools that we have in information theory and directly study natural communication. So there are biophysicists who do a lot of work in this area. There's a principle that's been articulated by at least some of them that basically states the following. If an organism has a communication task, so in other words, it needs to tell its neighbors something, or it needs to learn something from the environment, a sensing task basically, it stands to reason that it would have to do that as efficiently as possible. So evolution would likely select the organism that does that task as efficiently as possible, to use its resources efficiently. Information theory gives us a measure of how mathematically efficient a communication system can possibly be. So the principle is this: is it that evolution has discovered information theoretic limits, and therefore if we study molecular communication and apply information theory to it, can we make predictions about biological systems based on the mathematical analysis of information theory? So that's a pretty big and active area of research, both in engineering and in biophysics, and that's something I'm going to talk about a little later. And finally, so we talked about nano-networking, we talked about biology. Both of these were kind of like nanoscale ideas. It's also quite possible to have what are called wave-denied environments. So this is like at normal human scales, it's possible that you will be unable to send an electromagnetic wave or an electrical signal. So there are examples on this slide. We see underwater, so under seawater, it's not very easy to send electromagnetic radiation. Underground, in environments like heavy urban search and rescue, it's very difficult to send electromagnetic waves. So molecular communication has been proposed as a solution to just communicating at regular, normal human scales, just in places where radio doesn't work very well. And in fact, if you think about, so currently your cell phone, the most advanced cell phones are doing 5G. Some researchers are currently thinking about 6G, and molecular communication has been proposed as a 6G technology. Okay, so we've talked a little bit about what it is. We've talked a little bit about why we do it. Now let's talk a little bit about how it works. So consider the problem of sending a digital signal. So basically what you have is you have a transmitter on one side, so your cell phone. You have a receiver on the other side, that's like somebody else's cell phone. And what you're trying to do is you're trying to send a digital message from the transmitter to the receiver. So a digital message can be represented by a sequence of bits, such as this. This is just a random sequence. But in order to send it, it will clearly need to generate some physical process that is different from some other physical process. In other words, there's going to have to be a distinction that a receiver can draw so that a 0 and a 1 will look different, so the receiver can detect the difference between them. So in electromagnetic communication, this is very clear. You can have a high voltage for a 1, a low voltage for a 0, or a strong electric field for a 1, low electric field for a 0, something like that. There's reams and reams of theory about that, but you can easily imagine that there are differences that you can generate. So the question is, how does this work in chemistry? So how can you use chemical properties to send 0s and 1s? So there are really 3 methods. These are by no means the only possible methods, but these are the methods that have been discussed in the literature so far. If you're familiar at all with the communication literature, you'll recognize the word keying. That just basically means ways of transmitting 0s and 1s. And the techniques that we've learned of, or that we've developed so far, are called concentration shift keying, where you shift the magnitude, I guess, of the concentration. Molecule shift keying, where you alter the identity of the molecule that's being sent. And pulse position modulation, where you adjust the timing of the pulse of concentration that you're sending. So a quick idea of what these mean. So consider concentration shift keying. So what you're doing here is you're trying to generate a difference in terms of the concentration of whatever chemical it is you're trying to send. So roughly speaking, at the nanoscale, you're adjusting the number of molecules that you're trying to send. If you want to send a 0, so here in this example, oh, you can't see my cursor. In this example, you're trying to send a 0. So we're sending a 0. So the idea is you release no molecules. You do nothing, you release nothing. If you're trying to send a 1, you release lots of molecules. I should also mention in these diagrams, cell 1, let's think of that as the transmitter. Cell 2, let's think of that as the receiver. So cell 1 is going to release lots of molecules to send a 1. So this is kind of equivalent to an electrical bit where if you're trying to send a 0, you send 0 volts, and if you're trying to send a 1, you send like 5 volts or something like that. So this is basically very equivalent to that. And then what does the receiver do? So the receiver has to detect that difference in order to detect a bit. So what's the receiver going to do? It's going to measure the local concentration. Or at the nanoscale, it's basically to measure, it's going to count the number of molecules that arrive. So you can imagine, if we go back, if it releases no molecules, then none are going to arrive. So if it sees any molecules at all, it can probably conclude that a 1 was sent. It should also be clear from this diagram that there's noise, right? So some of these molecules are going to get lost. They'll disappear into the environment. So it is possible for you to transmit a large number of molecules. They all get lost, none arrive, and the receiver detects a 0 in error. So errors are very possible in this scenario. Molecule shift keying, that basically works by sending different identities of molecules, so different species of molecules, depending on what message you want to send. So the transmitter in this example, if it wants to send a 0, will release a molecule of type A, let's say a sodium ion. If it wants to send a 1, it'll release a molecule of type B, let's say that's a calcium ion. And then the job of the receiver is to measure the identity of the arrival. So in this case, a calcium ion arrived, so it detects a 1. Again, we can imagine the molecule will get lost, in which case it can't tell which one is sent. So this would be an example of what's called an erasure channel. In other words, you receive nothing and you have no information about what was actually sent. You know something was sent, but you have no idea what it was. So you're pretty much guaranteed to make an error in that scenario. Pulse position modulation. So this is the example where we use timing to send information. So in this example, the transmitter has a pulse of molecules that it wants to send. So it's got, like, for instance, a local sort of package of very high concentration molecules, and at the time that it chooses to send that information, it releases that, and so a pulse of molecules is released into the environment. So at the nanoscale, you can think of this as the transmitter has a molecule to release. It can release that molecule right now, but to send a 0. Or if it wants to send a 1, it can wait a bit and send it later. And then the receiver's job is to measure the arrival time of that molecule. In this scenario, so again, the molecule can get lost, but also, due to Brownian motion, that molecule is following a random path from the transmitter to the receiver. So, for example, the molecule that's released for a 0 might take an extremely circuitous path and arrive much, much later and be confused for a 1. So again, there's the possibility of error here. So errors sound bad, and they are, but they are where communication engineers like me make their money. So errors are the interesting part of communications, and that's basically what we study. Okay, so that's a bit, that is your 30,000-foot look at molecular communication. Now you know what it is, you know why it's important, and you know how it works. So I hope at least that sticks in your mind, if nothing else. But there's a lot more depth to it, and we'll start getting into some more mathematical models of it. So who knows who Claude Elwood Shannon is? Who does not? Okay. Quite often when I give this talk, every hand will go up when I ask who Claude Elwood Shannon is. So Claude Shannon is he's considered the father of information theory. He worked for Bell Labs, and in 1948 he wrote this paper, this seminal paper called The Mathematical Theory of Communication. And basically what this paper said, this paper made a number of contributions. Probably the two most important from our perspective are, for one thing, he said it is possible to send digital information arbitrarily reliably, so basically with practically 0 error across a noisy channel. So that's actually very interesting for us. We just talked a lot about errors. We talked a lot about noise. We have a noisy channel, so that's interesting. We can actually send reliable information. If you think about it, without that guarantee, all of the digital communication that you use in your daily life would be impossible. So imagine if you're doing your e-banking or whatever, and you do a transaction, and instead of sending $1,000, there's an error and you accidentally send $1 million. So that would be bad. The theory that Claude Shannon developed basically guaranteed that you can make it arbitrarily unlikely that you will ever make an error. In other words, you can't actually get to 0 technically, but you can make it so unlikely that in the age of the universe it would be impossible to make an error. The second thing that he did, and it's on this slide, he abstracted away the details of communication. So he came up with this very famous block diagram, which anyone who has ever done a digital communications course will have seen. And basically he said, okay, here are all the parts of a communication system. All you need to do is mathematically define them. So we start with a source of information that feeds information to a transmitter that produces a signal that can be sent across a channel. There's a noise source that produces distortion in that channel that results in the received signal. That received signal is observed by a receiver. The receiver processes that signal in order to try to figure out what the message is, and that estimated message is passed to the destination. So this seems very obvious, but it's actually quite profound, because you don't need to know, this isn't specific to any particular communication system. This applies just as well to your cell phone as it does to 2 cells in your body that are trying to communicate. And we can analyze them both using the same framework. So that's just a close-up view of the same thing, an abstract view of a communication system. So in other words, yeah, like if you think about things in terms of wireless, you're trying to send a message, you use your cell phone, you say hello into your cell phone, that is encoded, so there are various ways in which your voice is encoded by your cell phone. It goes through free space, there are various things that can corrupt it, noise, bandwidth restrictions, fading, and so on. These are all electromagnetic concepts, but at the end, the receiving cell phone that is your friend's will receive that signal, will decode it, and your friend will be able to hear your voice. So the insight is that it's exactly the same picture, except in the middle, in molecular communication, is exactly the same picture, except in the middle, the physical processes are different. So you consider your molecular communication cell phone. You say hello into it. It encodes your voice in various ways. We talked about ways in which you can encode information. Those molecules, patterns of molecules are released. There are various things that affect them, reactions, drift, diffusion. Ultimately, the receiver has to observe the effect of all those physical processes, decode the message, and their molecular communication cell phone will say into your ear, you'll hear your friend's voice saying hello. So it's the same idea, it's just different mathematical models. That's the only change. And in this slide, we can think about exactly where those would occur. So from the transmitter, what is the transmitter trying to do? It's trying to encode information. It's trying to encode binary information using chemical processes. So we can imagine that the signal would be generated in such a way as to be expressed in a fluid, so through diffusion in a fluid. How would we do that? Perhaps we would generate certain patterns of molecules, and we've talked about ways in which to do that. How does the noise arise? Well, the noise arises through diffusion. That's probably the single greatest source. There are other sources such as reactions and other things, but diffusion is the big one. We have mathematical models for diffusion. We can apply those directly to the noise source block in this diagram. And then the received signal represents the corruption of those patterns of molecules that we tried to transmit with the diffusion. So again, as long as we have mathematical models for all of these things, we can analyze molecular communication using information theory. The key quantities here, starting from the top, who has ever heard of mutual information? Very few people. Okay. It turns out that if I give you a mathematical model for a communication system, you can calculate that IXY thing at the top. I'm not going to get into the details, but basically that's the guaranteed rate. That's the rate at which you can transmit information reliably. There's a modification of that. The next line is called the capacity. That's your maximum over all the mutual informations. What you're maximizing over is basically all of the strategies that the transmitter can possibly produce. So this is the best you can possibly do, is the capacity. The last line, this actually comes up in biophysics a little bit. It's called the rate distortion, and it is applicable to scenarios in which you can't do some of the tricks you can do to calculate capacity, such as error control coding. But it again, the point is that it involves mutual information. So mutual information is a pretty important concept. Okay. Where do these mathematical models come from? So we talked at a very high level a little bit about it, but this is an abstract model of what a molecular communication system might look like. So the key issue is that the medium of transmission here involves Brownian motion. So what we're going to do is we're going to imagine that there's a point source of molecules here in the middle, and that's going to be my transmitter. So I'm going to imagine like a pipette that comes down from the ceiling into a medium, and just injects molecules right at that point, and those molecules don't, like the transmitter doesn't interact with the molecules after they're released, and the transmitter doesn't really have a volume. In other words, there's no self-interaction. So this is the easiest possible transmitter you could possibly imagine. The receiver, we're going to think of that as like a blob, right? A blob P. You can imagine like we're trying to send a message to a cell or something. We're trying to send a message to a bacteria. And so the receiver will be a blob over here. And the idea is that we're going to drop a molecule. We're going to send a signal by dropping a molecule, or maybe more than one, right at the transmitter. It's then going to, how is it going to propagate? It's going to propagate by Brownian motion or diffusion. So depending on how you look at it, it's either Brownian motion at the microscale or diffusion at the macroscale. And those molecules are going to propagate. The receiver has no idea what's going on until what happens, until that molecule arrives. So what's going to happen is, for each molecule, I'm going to get a release time, let's call it T, and that molecule is going to arrive sometime later. So in other words, that molecule is going to do its random walk until finally it encounters the boundary of the receiver. Let's assume that the receiver can perfectly measure when that particle arrives, And so what it's going to observe is that the molecule arrives at this later time, t plus n. So what's cool about that is that my signal in this instance is t, the timing of the release, and my observation is t plus n. So in other words, the first passage time of that Brownian motion becomes additive noise in this scenario. So now we have a mathematical model for our modulation. The way in which we're generating our signal is just a vector of release times. And we also have a mathematical model for our noise process, which is just a vector of first passage times. Often you can get the first passage time in closed form. Sometimes you can't, but often you can. And you can use that to analyze the system using information theoretic analysis. So here's an example. So imagine the following scenario. I have my point transmitter. I have a receiver in one dimension. So in other words, it's just a point in one dimension. And I have a Wiener process governing the Brownian motion of the particle, and there is net drift in the direction of the receiver. So this is one of the few scenarios in which you can actually solve the first passage time in closed form, and what you come up with is something called the inverse Gaussian distribution. So I've given you some examples here of the inverse Gaussian. It's governed by two parameters. One governs the average arrival time, and the other governs the intensity of the Brownian motion. But what you can see is that, for one thing, these are fairly broad-tailed distributions. So in other words, unlike a nice tight Gaussian distribution, the tail of this distribution is quite long, and that actually makes sense. If you think about Brownian motion, it's completely random, so the particle could take an arbitrary long sojourn in the wrong direction before it comes back. So the first arrival time can actually be quite long. As a result, we have what's called strong intersymbol interference. In other words, let's say I'm sending 1 molecule per second. So what I really want is I want that molecule to arrive before I send the next one. Because if there's 2 molecules that are propagating at the same time, I'm going to confuse them, which results in intersymbol interference. Unfortunately, with long propagation times, that either means I have to wait a ridiculously long time for that thing to arrive, or I have to risk the intersymbol interference. And normally what we do is we just risk it, because otherwise it's just too long to wait. Another thing that's really funny about molecular communication channels, and this might not be the first thing you think of, is that it's positive only. And why does that cause problems? So it turns out that there are many, many, many techniques, particularly in wireless, that rely on the fact that you can have positive and negative signals and they can cancel each other out. So in other words, there are reams of techniques that boost the bandwidth of like cell phone communication, for example, because what they can do is they can cause constructive interference, for example, in one direction, in the direction of your cell phone, and destructive interference everywhere else. In other words, everything else is canceling out, and that relies on the fact that it's positive and negative. Here, the arrival time, things like the arrival time, the concentration, things like that, they're all positive only. It's a non-coherent environment, so you can't get any cancellation except in very special cases, which we might talk about later. So as a result of all these problems, positive only, strong intersymbol interference, long propagation times, this is a very, very challenging environment for communication, which means as a communication engineer, I'm very excited. This is awesome. I will give you this quick result. I won't go through the derivation, but in the case of the additive inverse Gaussian channel, we've actually calculated not exactly the capacity, but bounds on the capacity. So about 10 years ago, we proved that the capacity is bounded between these two quantities, and H here is what's called the differential entropy of the inverse Gaussian distribution. So we've actually proven that you can actually calculate mutual information, or rather capacity, for these kinds of channels. So I think what I've talked about so far, so in terms of those like microscopic channels where you have like one molecule and it's traversing towards a very small receiver, and you can time things exactly, and you can count individual numbers of molecules. These are examples of like nano networking. So if you were trying to design a nanoscale network, these are the kinds of techniques that you would do. So that was like the first motivation that I had for molecular communication. So now we're going to talk about basically the other two motivations. And the first motivation, we'll give you a case study here. It's biological communication. In other words, communication between cells. How can we use molecular communication to analyze how cells communicate with each other? Okay, it's actually with the way signal transduction works. So signal transduction is the process by which your cells coordinate their activity. And what the picture is very similar to what I showed you. Basically, there are waves of concentration of certain ligands that cross the gap between cells, and those bind to receptors. So basically what we have is a picture that's very similar to what I showed you. One cell is producing a change in concentration or not to send some difference, to send some signal, and the receiving cell is trying to figure out what its neighboring cell sent in order to understand the message. So the only thing that's different here is terminology. Instead of like concentration shift keying, now I'm talking about signal transduction. And the molecules that traverse between the cells, we call them ligands. There are other ways to do that. We'll see examples. And in particular, our examples here are two receptors. One is called the CAMP receptor, the cyclic adenosine monophosphate receptor, which is a model organism, a model system for signal transduction that's found in slime mold amoeba. And another one called Channelrhodopsin. I'm going to show you this one because you can use the same techniques to analyze it, even though it actually isn't molecular. It's a light sensor, and it's a light-gated receptor found in algae. Okay, has anyone here heard of a dictyostelium? So there could be some biophysicists. I don't know. Are any biophysicists here? No. That's too bad. Okay. A dictyostelium is actually a really fascinating organism. So I just want to be 100% clear about this. I last took a formal biology course in grade 10. I don't have a wet lab, so everything that I'm telling you is stuff that I've read. And but I work with people who are very excited about this stuff, so they tell me a lot. Now I'm telling you. So I don't want to present myself as an expert in this, but it's a very cool system. So dictyostelium is the slime mold. That doesn't sound very exciting, but what it does is it switches between a unicellular phase and a multicellular phase. So for this reason, it's used, actually for a lot of reasons, it's used as a model organism, and that's one of them. It's a way to understand multicellularity. As we're going to see in a second, it's also used as a model organism because its receptor kinetics are very, very simple. So here's how it works. A slime mold is in a nutrient source. It's very happy. There's lots of food. It's eating, whatever. In that phase, it's unicellular. As soon as the food starts to run out, then it starts to go around that cycle. As soon as it detects the food starting to run out, it starts sending the CAMP signal. So it starts sending beacons, and what happens is the amoebas all start to come together. They come together in like agglomeration points. And once they do that, then they form like this weird spire. That's the picture on the right. And they form a little head on the end of that spire. And at the end of that head, a spore is formed. So basically, it's this agglomeration into a multicellular organization. Everything dies except the spore at the end. And so the spore waits until the food comes back, and then it repeats the cycle. So very neat system. I'm not sure if you can see that. Yeah, you should be able to see that. These are images of what are called CAMP waves that are developing. So initially, it's unorganized. This is actually not something that I study, but initially the CAMP is unorganized until, through a process that is actually poorly understood, certain beacon points are formed, and these beacon points generate waves of CAMP, and the other amoebas will converge on those beacon points to form the fruiting bodies. So you should be able to see waves that are sort of proceeding concentrically from beaconing points like that. So very cool system. What we're looking at is the operation of the individual receptor. These are ligand-gated channels. What I mean by that is basically that a molecule has to arrive in the receptor and then something happens. So the molecules out here represent the CAMP. What will happen is they will interact with a receptor. So on the surface of the cell, these orange guys are the receptors. So those CAMPs will interact with the receptor. Once they're bound to the CAMP, what happens is they produce second messengers, which cause the cell to take action. In this case, they'll force it to move towards the agglomeration point or do something else similar. Then once that's processed, the ligand is either absorbed or released. I'm actually not sure which, but in either case, the receptor unbinds and is now ready to receive another ligand like this. And that happens all over the cell. The other thing we're going to talk about is a receptor called Channelrhodopsin. Okay, I could ask this. Has anyone ever heard of optogenetics? A few people. So optogenetics is a very hot technique in neurology. Basically what you do, and by the way, as a theorist, I consider a lot of what biological labs do to be mad science. And this is an example. So like what they do is they take, somebody figured out the way you can do is genetically modify neurons in mammals, actually in any creature. And what you can do is you can genetically modify them so that they have this receptor, this Channelrhodopsin. It normally occurs in algae, but you can get it to express in neurons. Then what you do is it's light gated, so you hit it with a laser. What happens is it's an ion channel. The ion channel opens. A bunch of ions rush into the neuron, and then what happens? The neuron fires. So what you can do is you can basically use a laser to cause a nerve to fire. And there are kind of ridiculous experiments that I've seen. Actually, you can order a kit that'll do this, where you have, I think it's nematode worms or something like that. And basically what you do is you grow these nematode worms that are genetically modified like this. You hit them with a certain color of light, and you can get them to turn left. And you hit them with another color of light, and you can get them to turn right, which is just like poor worm, but very, very cool experiment. Anyway, we're not concerned with that. We're just concerned with the dynamics of what the Channelrhodopsin is doing. So in a light-gated channel, you hit it with a photon, not a ligand, but a photon. It opens, it emits an ion current like this. And that can happen all over the cell. Okay, so the key insight for modeling this mathematically is that these systems can be modeled as a Poisson process or as a Markov process. So that's great for me. I know a lot about Markov processes. They're very simple mathematical structures. You've probably heard of them. What it means is that there's a principle called mass action kinetics that suggests that there's a rate at which, for example, the CAMP receptor will go from unbound to bound and back to unbound. So those are two states of the Markov chains. You'll start in unbound. There's a Markov process that governs how it transits to the state of bound and back. So going from unbound to bound, the rate is governed by the concentration. That makes sense. The more molecules, the more likely it is to bound. Turns out that the unbinding process is independent of concentration. So that's kind of neat. You'll have in one direction, going in one direction, things are fast or slow depending on the concentration. So you can imagine how you can use that to send information. The complication is that to come back to the state where you're sensitive, that is actually independent of the concentration. So there's really nothing you can do about that. You can draw a state diagram that looks like this, going from unbound to bound. Dependent on concentration in the forward direction and independent in the reverse direction. So what we do in this analysis, you may have heard of this, we write a rate matrix that we put all the rates in. At any coordinate of that rate matrix, you have the rate going from, for example, unbound to bound, that would be alpha c, or bound to unbound, that's beta. And it turns out the way these rate matrices work, it's like a flux, so all the rows have to sum to 0 because the net flux is 0. The net flux in and out of the system is 0. And then we write what's called a master equation. It's just a vector differential equation that looks like what I have on the second row there. And that's basically how we analyze these. In fact, what I normally do is I discretize this in time, and we analyze them using Markov, mathematical techniques from Markov chains. I don't have enough time to get into the details, but they're frankly not that difficult, and we can certainly talk about them in the focus session afterwards. Channelrhodopsin, very simple. The only difference is, for one thing, it's photons. The illumination intensity now governs how quickly that opens. The other thing about channelrhodopsin is now there's 3 states. So it turns out there's a desensitized state after it closes, and then before it's ready to accept another photon, it has to pass through that degraded state into what's called the closed state. And now it comes back and it's ready to accept another photon. So the channelrhodopsin has 3 states: closed, open, and degraded. And basically you have to go all the way around the cycle in order to be sensitive again. You have to go all the way back to closed. But again, it's a Markov chain. It's just a 3-state Markov chain in this example. And the analysis is very, very similar. And there's the rate matrix. So again, you can write down the rate matrix. The takeaway message here is, if you know how to write down the rate matrix, basically the problem, well, the problem isn't quite done, but you're about 99% of the way through solving it. Okay, so some of the things that we've done with this, we've proven that if you want to calculate the Shannon capacity, you want to know how much information this channel can transduce. For one thing, we proved that the capacity is achieved with inputs only. So if you're allowed a range of inputs from low to high, then the best thing you can do is only use the lowest possible and the highest possible, nothing in between, and move as fast as possible from low to high. We were a bit confused about this. That's not really how diffusion works. One thing that we can say is that there are actually some physical systems that work like this. So for example, in synapses, it's known that the neurotransmitter, they're released in a huge blast, and then there are enzymes that try to clean it up as quickly as possible. So it seems to be trying to move from low to high to low again as quickly as possible. So this is a possible explanation for what's happening. We are also able to calculate the capacity of most of these kinds of systems in not quite closed form, but very close. So this is the calculation of capacity for the CAMP receptor. Another thing we were able to prove is that the channel capacity is equal to what's called the IID capacity when the input is IID. And it's also equal to the feedback capacity, which means that if you have feedback in the system, it actually doesn't help. And this is the expression for channelrhodopsin. So again, we can, in the focus session, I'd be very happy to go over the derivation, but it's not really important here, only that we actually can calculate the capacity in closed form. More generally, this with various definitions, we can express the capacity of a single receptor like this. Now, you might be interested to know how fast can this communication happen. So we're talking a lot about Shannon. We're trying to understand how quickly biological receptors can transduce information. And we came up with, so we plugged in the parameters that we know for channelrhodopsin. We came up with a number that I find surprisingly high. So this is, remember, this is a single protein that's sitting on the surface of a cell. So this is a very, very small system. We came up with the idea that channelrhodopsin at maximum, so at the peak of that figure, can transduce about 66 bits per second. Again, I thought that was quite high. If you just think about how fast things normally proceed in biology, they're not very fast. But now we're talking about like almost 100 bits per second for one single protein. We can also ask, so these receptors don't occur singly; they occur multiply, and we can ask, well, what's the potential information rate when you have many receptors? So we actually solved that as well, and we came up with, under some circumstances, it scales linearly. So in other words, if you have n receptors, then the capacity is n times as much. So on a typical algae, you might have, if you have 1 million receptors, which is not unreasonable, they're just single proteins. If you have 1 million receptors, 66 bits per second per receptor, now you're talking about some serious information rates. We're talking about 66 megabits per second. I like to call this biological MIMO. And MIMO stands for multiple input, multiple output. That's a term from wireless that indicates that you can get huge gains by adding the number of antennas. By the way, MIMO is why, if you have a Wi-Fi router at home and you're wondering why it has like 4 antennas, that's MIMO. So every antenna that you add basically increases the number of channels that you have and increases your rate. So this is an example of biological MIMO. So that's what we can think about as far as biology. I can talk, I want to move on to this last part. Again, in the focus session, we can talk a little bit about what the implications are of what I just said. But I do want to introduce to you my final case study, which is sort of a little more silly and a little more fun, which is tabletop molecular communication. So this was, again, when I was first starting to think about molecular communication, a lot of people were thinking about this as a nanoscale technique, or certainly a technique in which you needed a big complicated wet lab to get any results at all. So there I was sitting, a fairly junior faculty member with fairly small grants, and I was thinking, well, I'm never going to get $1 million to get a wet lab, and even if someone was foolish enough to give me that money, I wouldn't know what to do with it. I've never operated in a wet lab. So I was sitting around with a colleague and one of my grad students who were just bouncing ideas around, and I remember we were thinking, well, you know, what kind of chemical sensors can we play with? And actually the first thing that my colleague said was, well, what about a breathalyzer? And we were like, yeah, okay, that would work. Like you could, if you drink a bottle of vodka and then breathe on a breathalyzer and then don't breathe, you can send either a 1 or a 0. So that was literally the genesis of this idea. But really, this is the thing that we actually built. But really, this is a potential solution, like I said, to the wave-denied problem, right? Like if there's an environment in which you're trying to send information, but you can't use electromagnetic information, you could potentially produce information in puffs of molecules in free space diffusion. Anyway, like I said, this is the device that we actually built. On the left-hand side, you can see that is, we went out and got a motorized spray gun. So, you know, Windex bottles, basically. This is just a motorized version of that. And my grad student modified it so that the motor was controlled by an Arduino. So basically he could, in order to send a 1, like he activated the motor so it sprayed a puff, and in order to send a 0, he did nothing, so it didn't do anything. On the right-hand side, it's a little hard to see, it's a little blurry, but that's the sensor. So again, basically what we did, we abandoned the breathalyzer idea pretty quickly, but what we did was we went to various websites and we just saw what kind of alcohol sensors we could get, and we actually built a device with a bunch of different ones because we weren't exactly sure which one was going to work best. And again, that's connected to Arduino. The computer in the middle, it looks like we're cheating. It looks like it's connected on both sides, but no, that's just there to do the signal processing from the receiver. That's a close-up of the receiver that we built. Again, this was like, I think we spent $500, and I think that was like learning everything we needed to learn and making mistakes. I think knowing everything we know now, probably all the materials to build a new one would be like $100. So like any of you could go out and do this if you wanted. Like I said, we put 3 sensors on it because we weren't sure which one was going to work best. I think it was the MQ3, the one in the middle there, that ended up working the best and the one that we used, but it was all controlled by Arduino. Okay, what did we find? So on the left, this is the response to a single spray. So this is as though we're sending a 1, and this is what happens in the environment when we send a 1. This is 5 separate trials. So in other words, what my grad student did was he took 5 measurements where he would spray, then wait for it to dissipate, then he would spray again and lined them all up. As you can see, there's actually quite a bit of variation between trials. So that is something that is relatively unusual in communication, is that in this case, the transmitter is actually quite noisy. So we don't have a lot of consistent behavior from the transmitter. On the right-hand side, we see an example from one of the communication runs that he did. And basically, at the top of that curve, you see the information sequence that was sent. So anytime you see a 1, a puff of alcohol was produced, and anytime you see a 0, nothing was done. if you look at that, you can sort of understand that, the signals are really interfering with each other quite strongly. So in other words, the more alcohol that's already in the system, it just sort of builds up and builds up and builds up, and so you get this sort of curve that's constantly increasing. That required quite a bit of thinking and signal processing in order to actually understand what the signal was based on what we received. But ultimately, my grad student actually figured out a pretty clever algorithm, and we were able to decode most of these sequences without errors. This was, as you can imagine, this paper, we published it in PLOS One. It dropped around Christmas time, and we told our media office, we figured out a way to communicate with vodka. And they thought this was hilarious, and they wrote a press release, and because it landed around Christmas time, everyone thought this was really funny, so we ended up getting in a lot of papers. But a pretty good question that I hope is occurring to you is, well, how much information could we actually get out of it? And the answer is not very much. So about a bit every 3 seconds. So this is more like sort of what we were expecting. So we saw the biology, the individual receptor. You could get like almost 100 bits per second. This is like much closer to what we were thinking. It's like, yeah, this isn't very fast at all. You're not going to be downloading movies over this channel. But so some people, including ourselves and some other groups, have sort of taken this idea and developed it further. I, along with a group in South Korea, we actually did implement a molecular MIMO type environment where we had 2 transmitters and 2 receivers. And this is what it looked like. This is in their lab in Seoul, South Korea. On the right, you can see some of the signals that were measured. So basically, the idea is you have 2 transmitters, 2 receivers, so you can send independent streams of data. They'll interfere with each other, but you can actually use the interference to help with the signal processing. And here, we were actually able to boost by almost an order of magnitude to about 2 bits per second. So again, not a huge amount, but there's improvement. Subsequently, my former grad student, who's now at Toronto Metropolitan as a professor, understood that a big problem here, like I said, is non-negativity. And his idea was, well, where can we get a system that introduces a kind of negativity? And his insight was, well, if you transmit signals using acids and bases, they can be used to cancel each other out. And that's what he did. He designed a similar system to us. It was all in fluids because it's acids and bases, but he was able to get 3 bits per second. I just read of, and I think this is the world record at this point, I just read of a system that was designed by a group at Brownian University, by a guy I know very well there, and they were able to get, I think, up to 20 bits per second. So this is continuing to move, and we're continuing to be able to get higher and higher information rates using these kinds of techniques. And I think that high-speed molecular communication is possible, and we'll start moving in those directions any day now. Okay, so that basically wraps up what I wanted to talk about today. And I hope you take away the following ideas. Firstly, is molecular communication a viable form of communication? It clearly is. So we can transmit information. It seems like we're able to transmit increasing rates. There's no real clear limit on the kinds of techniques we can use to improve it. Furthermore, can molecular communication help us understand biology? Definitely. So we've done a lot of analysis, and there are ways in which this can actually influence biophysical analysis of systems. And again, we can talk about that a bit in the focus session if you're interested. And the final thing to take away is there more work to do? Of course there is. There's tons of work, and we're continuing to do some. I hope I've inspired you to maybe do a bit yourself, design some tabletop system very inexpensively using off-the-shelf components, and try a bit of molecular communication for yourself. So that's all I have to say, and thank you very much.